Good afternoon. Today I will be presenting on porin protein. First, I would like to introduce proteins. Proteins could be considered the building blocks of life, since they have many different functions in both prokaryotic and eukaryotic organisms. Some of the functions in humans, which are eukaryotic organisms, includes vision, motility, and transportation of molecules from cell to cell. Proteins have four levels of structure: primary structure, secondary structure, tertiary structure, and quaternary structure. Primary structure is the amino acid sequence determined by the DNA of the protein. The secondary structure is based on the primary structure and is the repeated foldings of the polypeptide chain, which is the amino acid sequence, into either beta sheets or alpha helices. Tertiary structures are the 3D conformation of the entire polypeptide chain. All proteins have primary, secondary, tertiary structures, but only some proteins have quaternary structures. Quaternary structures are the is when there are more than one polypeptide chain present and it's binded to another entire polypeptide chain through covalent bonds. Porin proteins function mainly as a transport channel. It transports ions through the outer membranes of mitochondria and gram-negative bacteria. There are two types of porin proteins. One is general diffusion proteins, which diffuse small hydrophilic molecules across membranes and are generally not selective, whereas selective porin proteins have a binding site and are moderately selective of the molecules that they transport. These porin proteins have primary structure, secondary structure, and tertiary structure. In the picture here, we can see the tertiary structure and the secondary structure of porin proteins. Here we can see the alpha helices, and here we can see the beta sheets, which is the secondary structure. As a whole, this entire thing is a beta barrel, which is its tertiary structure. It is the 3D conformation of porin protein. This one here is a monomer, since it's just one. Now here we have the quaternary structure of porin protein. As you can see, there are 3 polypeptide chains folded into beta barrels. This entire structure can be called a trimer. The DNA sequence of porin proteins is in this slide. It is GGT, GGT, CT, GGT, G, CAA, TA, GG, CCC, TAA, CTT, CCC, ACC, AGGA, GGT. There are multiple diseases associated with porin protein. Some of them include antibiotic resistance, type 2 diabetes, genetic cataracts, fat accumulation, and brucellosis. Antibiotic resistance occurs due to the fact that porin proteins are commonly found in gram-negative bacteria. A study was done in order to find the effects of porin protein on antibiotic resistance. It found that since porins are also found in gram-negative bacteria, porins increase antibiotic resistance. Finally, to conclude this presentation, proteins are a very critical part of the cells of prokaryotic and eukaryotic organisms. Porin proteins can have both positive and negative impacts on an organism. And these are my citations.